Welcome to the first TMDA 2021 meeting. For medical device packaging and validation, there's more to it than you think. We're going to quickly cover a couple of housekeeping issues, which is please place your computer and cell phone on mute and cameras off. If you're using your phone, make sure to mute it with your computer. At any time, please use the chat box. So this is where we're going to get our questions during the question and answer session. And this webinar will be recorded and placed on the TMDA website. So we're very fortunate today to have 4 experts with us covering various aspects of medical device packaging. Each of the panelists have extensive packaging experience, ranging from design, manufacturing, validation, and testing. To get started, I'd like to turn the meeting over to each of the panelists to provide a short background on themselves. We have Dana Wilcox, Andrew Fronk, Andrew Cockerham, and Jennifer Gygi. Do you want to go ahead, Dana?
Hi, thank you for the introduction. My name is Dana Wilcox. I'm the CEO of Mainstream Medical Devices here in Austin, Texas. We are providing contract and sterilization services for startup, small and dynamic product lines. I've been in the medical device industry for more than 20 years and look forward to our discussion today.
And this is Andy Fronk. I am a medical device packaging engineer and a consultant with Bear Creek, physically located in San Antonio, Texas. So I went through the storm as well as you guys did up in Austin. I've got about between 25 and 30 years of medical device experience, a little bit of consumer products experience sprinkled in early in my career. For the last 10 years, I've been consulting with a lot of startup companies, medium-sized companies in the area of medical packaging, especially sterile packaging, as it has a lot of regulatory requirements. So that's my background.
Hi, everyone. I'm Andrew Cockrum. I'm Director of Quality at Alafair Biosciences here in Austin. I have about 10 years experience in medical device in startups. My first startup from early R&D through acquisition, and now here with Alafair, and definitely excited to talk about packaging and not talk about whether I have power or water.
Amen.
My name is Jennifer Gygi. I work at Nelson Labs. We're based in Salt Lake City, but we have facilities all around the country and the world. We're part of a larger company called Sotera Health, which includes our lab, Nelson Labs, Sterigenics, which is sterilization services, and then Nordion, which provides our Cobalt 60. Nelson Labs specializes in medical device testing, and we do some pharmaceutical. I've been here for almost 27 years, next year. And I've moved around, done a lot of different groups here. So a lot of exposure to different types of microbiological tests. But I spent 15 years in the packaging group and was instrumental in helping to get that group up and running. And so I have a lot of packaging expertise. Right now, I'm currently working in the technical consulting services division of the lab.
Well, thank you, all of you. Next, what we are going to do is I'm going to ask some questions and ask panelists to answer those questions. So let's go ahead and get started with Andrew Cockerham. How do people include customer requirements in the packaging design?
Yeah, that's a great question. I think people often, wonder, well, how early do I bring packaging design into my product development process? And it really goes all the way back to the beginning from a customer requirement. So hopefully you've already started with a customer requirement that's related to packaging, and maybe several of them. For example, it could be as simple as packaging is easy to use. Obviously, then you have sterilization and other things that can come into play. So once you start with that requirement to make sure that it's included early in the process, there's a couple different ways to include the customer in that design process. So surveys are one helpful way to get feedback from surgeons early, and hopefully you're doing that anyway for the design of the device. You have physicians involved, so getting their input on packaging is something that can be forgotten, but is very helpful. And then also, I think remembering that the customer for the packaging is definitely the surgeon or the end user, but also it might be the surgical tech, right? Or the other OR personnel that are the ones actually opening the package, at least if you're in a sterile, sterile environment, double pouch situation, often it's not the actual surgeon opening the packaging, it's the tech. So getting those people, getting input from those type of people on what is easy to use, what works, and what they're used to. So also looking at other devices that might be similar to your device and looking at what their packaging looks like. Is it a tray? Is it a pouch? So getting all of that feedback early on, I think will ensure that you don't have packaging issues coming from your customers later on. And then following on to that, I think when you get to the point where you have your design validation, you're also doing cadaver labs, probably, or other types of design validation for those customer requirements. So again, you're bringing in users and having them evaluate the packaging at that point. Also, hopefully at that point, you don't have to make any additional changes, but you're getting their input of, yes, this packaging meets my needs, this packaging will work.
I guess we have to be quiet now. But I was wanting you to see if you could look at that.
Follow on with that question, I thought I would ask Andy, what are the regulatory regulations covering sterile packaging?
Can everybody go on mute, please?
Yeah, good question. The main one that I think most people in medical device are aware of or should be aware of is ISO 11607. There's a 2020 version that just came out last year. Basically, what that walks you through is all the validations. It's not super prescriptive, but it's fairly detailed. The validations that are involved for the packaging itself, design inputs, talks about specifications, it talks about vendor requirements, packaging vendor requirements for some of their validations. And that's all encompassed in part one. Part one also includes packaging shelf life, as well as performance testing. So that would be your shipping testing, that would be sterilization, compatibility, and things of that nature. And then 11607 part two is having to deal with the forming, if you have like a form fill seal type product, or the sealing, which a lot of us are familiar with for, say, like a pouch or a tray system. It deals with the validations for those, and requirements for the forming and sealing instruments to be also validated. So they're IQ'd and then OQ'd, PQ type process validation. So it goes through that. So that's probably the really critical one. If you understand 11607 and you are compliant with it, that's probably the biggest piece that you have to tackle with a sterile medical device.
Thank you. I thought maybe, Dana, you could help people by telling people how to start a company, what kind of contract packager could they look for?
Sure, I'd be happy to answer that question. Really, with any startup, small or dynamic product line, you're looking for a contract packager that can help you work through all the steps and some of the regulations like Andy was just describing, walking through doing packaging validation, shipping testing, and then as your actual contract packager, once those things are developed and set, it's helpful for the packager to be flexible, to be more of a partner, to be able to walk through issues with you as a startup company, because as Andrew was talking about, you have customer requirements at the beginning, but they kind of get sorted out as you work through your processes. So having someone come alongside you and work through those issues. I'd say some really important steps for contract packagers or to be able to provide you with cleaning and cleaning validation, including reports and protocols, and help you with those steps. And then the sterility validation, being able to provide you guidance on bioburden and how to leverage the standardization of actually the dose that's required for the sterility validation. And again, with reports and protocols. And then finally, if they have the option to adopt you into their product family for sterility validation, that's also a really great step that helps startup and small companies, saves them time and money, and really helps with sending out potentially later gamma runs that can be shared.
Thank you, Dana. On a related note, Jennifer, perhaps you could talk about an overview of the test requirements for a new package design.
Certainly. If you're validating packaging, you're going to be following that ISO 11607 document that has been talked about. And essentially what you're looking to show is that your package starts out in a good condition, and that after aging at the end of its life, it's still going to maintain that good condition. And the way that they recommend doing that is using a three-pronged approach where you're looking at strength, integrity, and microbial barrier attributes of the packaging. This should be done under worst case conditions, things like maybe a 2x sterilization. Maybe you want to seal your packages at the lower setting instead of the nominal setting. So you have worst case sealing, worst case sterilization. That way, you know that you have a lot of confidence that whatever your validation data shows, if it shows that your packaging is acceptable, you have a lot of confidence that that data is going to be good no matter what, because in the worst possible instance, your package was still passing. And we would also do it in your final packaging with your product or something very closely that simulates it. And then you would do a distribution simulation to kind of really challenge the packaging and mimic what it's going to be seeing possibly in the field. Then you'd run those strength, integrity, and microbial barrier properties tests on them. Then you would age the product. Accelerated aging is very often done to get data quicker so that you can get to market faster, but you are required to start a real-time evaluation and do that as well. Once you've got the product aged to its final time point, you repeat the same test that you did on it in the baseline condition, and hopefully you've met your acceptance criteria that you established at the beginning of the validation, and you've shown that your package is acceptable.
Thank you, Jennifer. I thought maybe, Andy, you could tell what some trends in packaging problems might be.
Oh boy, can of worms. So I have seen a couple interesting things lately with some of my clients. Obviously, I can't say who they are, but I see a lot of different things. One of them has been an interesting phenomenon with actually with glove contamination. So everybody wears, nitrile gloves or neoprene gloves or some sort of a glove rather than bare hands to keep microbial loads down. But there's a byproduct on the gloves that is interfering with certain kinds of Tyvek lid or paper lid adhesives. So it's causing some integrity issues, some visual anomalies. I've not seen an actual leaker as a result of this, but I've seen some stuff that's really close and visually unacceptable, like when you do a peel back on a tray, for example. So it's I believe it's calcium stearate that's the contamination, and I don't know if it's because of kind of COVID and things that people are producing more gloves faster or something, but we're seeing this more than we've seen before with from a couple of my customers actually. So just something to be aware of if you're already doing packaging out there. And then a second thing that I'm noticing, remember when DuPont introduced their transition Tyvek several years ago, and they did a lot of round robin testing to show that it was the equivalent or better. And I think for the most part, that's the case, but A lot of people portrayed it as just a direct plug-in without doing a lot of testing. And I am seeing some cases recently where the transition Tyvek is behaving a little differently with sealing validations and sealing processes that have already been established. For example, they're having to use a little bit more heat or a little bit higher dwell times to get the same quality of seal as before. So I don't know if this is transition Tyvek as much as it just maybe is an anomaly at DuPont a little bit right now. So I'm seeing those two things just as a current trend, current issues.
Thank you. Jennifer, I have a question for you. Do I have to repeat the validation if I make a change to the packaging?
That's a question we get asked a lot. And the answer is it depends. Anytime that you're making a change, you need to do an evaluation as to what impact that change may have. And most of this is really covered by your company's change control procedures. And you would evaluate whether or not that change is going to have an impact. If you decide that it doesn't really have an impact on your packaging, then you can usually write a paper justification in your change control saying, you know, this, we've evaluated it, we looked at it, it's fine. If it's got a potential impact, maybe it's somewhere in the middle, then we would suggest maybe doing some kind of confirmatory type testing, you know, do a few things just to make sure that that change doesn't really have an impact. And then sometimes it's very obvious that that change is going to be impactful. And in those cases, you may want to repeat your entire validation. So it's really going to depend. And another thing that we try to push out there is that you need to not just evaluate every change by itself, but you need to go back periodically and review all the changes, because a lot of times very small changes that in and of themselves didn't make an impact to your packaging, cumulatively, they may start making an impact. So it's important to go back and look at that in a more holistic approach.
Yeah, and I'll add just a little bit to that. I think, you know, if you say redo your packaging validation, you know, as we've alluded to, there are several components to packaging validation. There's the sterilization, there's the aging, there's the product testing inside the packaging. So it would probably be unusual for you to have to redo all of that work for a packaging change, although possible, but it might be likely that you would redo one portion of that testing. So, you know, for example, if the, you know, the box, the outer carton that you were changing wanted to go for a different size or something, you may not have to redo all portions of your packaging, but you might redo distribution simulation or something like that, because you could write a rationale that that was the only part of the packaging validation affected by that change. So it really just depends on what the change is and assessing how that change impacts the testing that you've done, and then what testing would maybe be repeated or done on the new design.
Yeah, so it's essentially just a case by case that needs to be evaluated.
I have a related question also. This is specific to the package testing. And it says, is dye leak or bubble leak a better test method for determining package integrity?
I guess my answer to that, this is Andy, is that they're both good methods, and they're both kind of part of the arsenal of tests that you'd want to use. Dye leak is very good for sealing process validations, where you're looking at the interface between a Tyvek lid, say, and the tray, or both sides of the pouch being fused to form a sealed package. It's an excellent way to look for those kind of leaks. Bubble leak is better for whole package integrity, say post distribution simulation, to look for, you know, pinholes and creases. It's something you'd want to do post aging, post shelf life, to look for pinholes in the where a pouch was bent and folded over time. It created a pinhole, or again, post distribution, where you have shock and vibration that might have torn your package or burst a seal. It's a little bit better for that. So they're really both very useful and necessary test methods.
Perfect, thank you. So I was going to see, Jennifer, could you tell us a little bit about material and sterilization method compatibility? I get asked that question quite frequently.
Well, when you're designing your device, you need to be thinking about sterilization, right? Because some of these design elements that you put in may preclude one type of sterilization or another. And the same thing really goes for packaging, not quite to the same extent, because there's great packaging solutions, whether you're, all the sterilization methods that are out there, there's multiple packaging solutions that are compatible with those. But you do need to think about some of your materials compatibility, because not everything's compatible with radiation. A lot of materials are going to become brittle. They're going to lose some of their strength. They may experience some color changes. You may run into some issues with some materials with EO, not as much with compatibility, but more with the breathability in order to get the ethylene oxide into the product if you're using a gas sterilization. So it is something to think about, especially if you're trying to do some kind of a product grouping. Like Dana was talking about, if you want to get into a group, you need to be similar in that group as a whole. So you would want to have the same packaging type if you wanted to become part of a family, because the more similar your packaging is, the easier argument it is to make that you belong together in the family and that you don't belong in your own family.
So talking about sterilization and sterilization families, I was wondering if, Dana, you could talk a little bit about how dose mapping relates to sterility validation.
Sure. So dose mapping is actually a separate step. It's based on your sterility validation information and dosing that you've established as part of your standardization. The dose mapping actually happens at the sterilization vendor, and the vendor themselves are responsible for doing the triplicate dose map to make sure that the dose is effectively consistent over your product and in the overshipper. So really they happen at two different times. You have to do the sterility validation first to establish your low dose range and the dose range for the product. And if you're part of a product family, then they run all in the same range. And then the second step to that is really performing the dose mapping at the sterilization vendor. So sterility validation is more micro, dose mapping is more about how well the concentration of the radiation is going to your product.
Thank you. So I have another question. I'm wondering if every time we get a package, do we have to validate the total package, each and every configuration for package validation, or can we just make a worst case?
What I like to do is make a worst case. I think that's the best strategy. As long as you have established a packaging family, you can test the worst case product in that family. It may not necessarily always be the obvious choice. It could be the heaviest, but it also could be a smaller unit too. It could be the largest, could be the smallest. The smallest may move around more. That could be another hazard. The largest one obviously could burst seals and things like that. So you have to be careful with your rationale, but I think you don't have to test every iteration of a product generally if you have a packaging family established. There's ways around doing that.
Agreed. But then what happens if the packaging comes in and it fails at receiving inspection? Andrew?
Yeah, well, hopefully all of your failures or nonconformances happen when the product has not been distributed yet. It's still in your control. Of course, you'd quarantine it. Then you do a root cause evaluation and assessment of what happened, and try to understand what the failure is and what it impacts. So, for example, if the failure might be that the material is different. For example, the outer shipper carton, if it's a different thickness, then that would impact certain things, but not other things. So you have to look: does that impact the sterility? Is there a chance that this product is not sterile? But so if the answer is yes, then it's a bigger issue. If the answer is no, well, then is this going to impact my temperature, environmental conditioning? Is this going to impact my distribution simulation? So you would obviously hope to retain the product and not have to scrap it. So you could do retesting of whatever the failure is, and try to get data to understand, okay, maybe I can use this product, but I got to go do some testing on what failed to be able to save the product. So once you know what the failure is, then you can assess what is the impact, and then you don't have to automatically throw away all the product. You can gather a body of evidence, maybe it's additional data, to determine what maybe the product is acceptable in some different configuration based on your testing. And some of it's a little bit of a case by case, of course, because some testing, like accelerated aging, takes a long time. So if what failed might impact aging, then it's going to be a little bit of a turnaround to get data. But some testing is not very long. So distribution simulation testing, you can get that data in a week or two. So depending on what is impacted, you may be able to get a data set to support packaging that isn't what you validated, but you can get information to support it in a case of a failure.
Thank you. I have another question, only this time it's about microbial testing. And does microbial testing need to be part of the validation test plan? Jennifer?
Yeah, I'll take that one. We do get asked this one quite a bit at the lab, and it is listed in the 11607 standard. So you need to incorporate that as part of your validation. Most of the time, we're just telling people to just run the test, because again, you're weighing the costs of rationalizing not doing it or getting that data from another means versus just the cost of testing. And usually it's about the same, so it's easier to just do the testing. But if you're using, let's say, DuPont's Tyvek, right, they have published a lot of data and they've done a lot of testing on it, and that testing includes microbial testing. So can you leverage some of that data that the supplier has given you and write a justification saying this is what this material is that I purchased? I would say probably yes. Most of the time, we just advise people, again, to do the testing, because kind of from a cost perspective, the time and effort taken to develop the document showing that the manufacturer's information and how you're leveraging that may be about the same when all is said and done. So I would say yes, you should include the microbial because it's in the standard, but does it mean that you have to do testing? Not necessarily.
Thank you. I was also wondering is if you use a contract manufacturer, do I need to worry about process validation? So if you contract that out, is that the responsibility of the contract manufacturer or of the manufacturers themselves? Dana?
Yeah, I can take that one. So oftentimes contract manufacturers or packagers have pre-validated pouches or some kind of product that they've already done all the sealing validation for and process validation in their facility. If you can leverage some of that pre-validated packaging, then that removes a lot of the cost. And it additionally, those activities don't have to be repeated. If you end up wanting to go with a custom, let's say, pouch, that the thickness layers are substantially different from their pre-validated pouch that they use, then you would likely have to do some kind of new process validation in conjunction with your contract packager. If they have to put in new equipment or some kind of new process, then yes, that would need to be done at that point.
Thank you. So I don't have more questions for the panels at this time. Should we go ahead and open it up for the questions that came in through the chat line? There's quite a few.
And that's a great idea. Thank you.
Great. I'm just going to start at the top. Okay, so we have someone who wants to link to ISO 11607. Specifically, they're interested primarily in the outside package as they don't have a sterile medical device. So how does 11607 relate to a non-sterile device?
I've been asked this before. It doesn't directly apply because 11607 is specifically for sterile, but to me, the principles still apply that you need to protect your product. You need to protect your device, whether it's sterile or non-sterile. So there are FDA and other requirements for making sure your product is being protected, that you can distribute it, that labeling integrity and legibility are maintained. Those types of things are still applicable for a non-sterile device as well. So some level of testing, it would not be as extensive. You're not doing integrity testing on a poly bag necessarily, but you would want to show that it can be distributed without damaging your device. So there are some requirements. It's just not 11607. There would be some other just general requirements of protective packaging on your product and labeling legibility. That's my thoughts on it.
Yeah,
Thank
that's well
you.
said, Andy. I'll just add really quick, so another thing that may be applicable that's not sterility might be temperature. So as Andy said, you got to protect your product and make sure that it's still going to perform. Distribution simulation and ship testing might be applicable if you have temperature requirements for your product that it performs under. So it just depends on your product and what the requirements are.
Yeah, it's a good point. Like diagnostics products may not be sterile, but they still have a lot of requirements, and you may want to use some of the elements of 11607, even, if you're packaging, a liquid or assay or control or something like that. So yeah, that's a good point.
Okay. And then the next question is, does ISO 11607 require that accelerated and real-time aging have to be started at the same time?
It actually does say they need to be started within 3 months. And I think that's in the newest version. And that was because they were finding that people were forgetting to do it because they didn't do it at the same time as their accelerated. So it does say within 3 months. And that is really the expectation.
That's interesting.
Yeah, that's in section 8.3.4.
Guess they got sick of finding it in audits, so they decided to address it.
Okay, next, someone asked, could you go over the selecting proper sample size for packaging validation and if there are any standards to guide you? Who wants to take that? Andrew?
Yeah.
I can start it off. I mean, to me, I think it's based on risk. I see that larger companies, they tend to have much larger sample sizes, and say for attribute data like pass fail, they go for 95, 99 sometimes confidence, reliability. I recommend, I like 60. That gets you to 59, which is gives you the 95, 95 for sterile barrier integrity for most purposes, for most companies. That tends to work, but it's really based on risk, and a lot of people have a lot of companies have separate procedures for statistical analysis that they will apply to that, and they'll have a separate kind of a risk plan based on what kind of harm, in the case of a package, what kind of harm can be done if it ends up being not sterile. Usually that's classified as fairly significant event. You could have a patient infection, those types of things for an implant, for example. So that usually drives high sample sizes for pass-fail things. For variable data like seal strength, for example, you can get by with much lower sample sizes and still have a statistically significant numbers to get PPK or CPK for process validations.
Yeah, that's well said, Andy. I think you go by the risk assessment that you have for your product. If it's sterile, as you said, that'd be a high risk, but it may not be a sterile product, so it depends on the risk of your product. So ISO 14971 would be the risk standard that would be helpful. And yeah, just to add on to Andy, 95 is pretty common, which is a sample size of 59 for smaller companies, and I know larger companies like to go 99 for a lot of things, so you get pretty high sample sizes. The only other thing I'll add is 59, if your goal of sample size is 59, it helps to use a few extra samples in there when you actually run the testing, because there might be test failures or other things, and you don't want to have like 2-year real-time aging and then have a test failure, and you only tested 59 and you had 2 more years to get another unit to test. So it's usually helpful to throw in a couple extra above the sample size that you want, just as a little bit of an insurance for test failures or other things that can happen.
That's a great point. Excellent.
We have another question related to one of the previous, and it's about if you have multiple sterilized pouched products using the same pouch materials but different geometries, do you have to do all of the testing, including real-time aging and performance testing, or on each configuration, or can you do the worst case configuration?
That's a complicated question. Sounds like it's a blend of several different things. I'll answer part of it. Maybe somebody else can jump in and answer the rest. But to me, for stability and shelf life, it's mostly material related. It's your packaging structure, and it's your sterilization method. And it's less about validating on a specific piece of sealing equipment. That's your process validations. So if you have a family established for a certain kind of pouch, it's based on that pouch material and the sterilization method mostly. Sometimes your product does interact over time, so you want to factor that in. You've got to have that be a consideration. But for stability, it's more about that material plus sterilization, and less about how big the pouch is or whatever. A lot of times you'll pick a larger tray system or a larger pouch system just so you have maybe more surface area, more sealed area that you can test over time. So some people like to bracket. So you'll have maybe the smallest pouch, the largest pouch. You'll kind of bracket everything in between. You don't have to do additional seal testing or additional stability testing on a system if you do the bracketing approach. So there's different ways to kind of handle that piece of it.
Yeah, as Andy said, it's definitely the standard includes worst case consideration. So you can follow the standard and use a worst case consideration, and it's industry standard to use for example, 1 pouch and seal settings as your worst case, rather than doing multiple, and you adopt them based on the worst case approach. But just a warning, so absolutely do that, but be careful and make sure you really have documented exactly what is worst case. It may not be so obvious what is worst case. And certainly, I've seen questions from the FDA during submissions about, no, we don't really agree that that's worst case. Maybe you need to go back and you may have to redo testing on something that you didn't do, or do testing on something you didn't test because you said it was worst case, but the FDA may not agree with your assessment of why that is worst case. So just make sure you have a good understanding and rationale for what is worst case, but it's certainly common to use a worst case approach when applicable.
And just along those same lines, I mean, when you're writing your protocols and you're describing your test plan, I mean, tell a good story. You need to have a good rationale, and that's a great place to put it, is explaining why something's worst case and explaining that it's weight, geometry, whatever. Go into detail there. It'll save you some time later on from being audited and have some finding where you didn't tell a good enough story.
Yeah, that's well said.
We have a question now about non-sterile packages. And so the question is, are there packaging or distribution test requirements for a non-sterile device?
So I can jump in. I'm not sure off the top of my head of a specific standard that calls out non-sterile device requirements, but I think the short answer is probably yes. In other words, you have to show that your device is going to perform as it's intended when it's used. So if you're ever going to ship your product to the user, then you would need to have some data to show that it's going to be performing as intended when it arrives. So again, it kind of depends on what you're doing and what your use case is. If you're selling something that's, you know, the size of a room that doesn't get shipped, it's like installed in pieces or something, you know, maybe you wouldn't need distribution testing. But if the product is going to be distributed through FedEx or some other shipping, you just got to show that it's not going to be damaged and your packaging protects it to the point of use.
May I address this?
Yeah, you wrote the answer, Mike. Just go ahead and read that part. That's fine.
Okay. I'm not a part of the panel. I do work for Andrew Fronk at Bear Creek Consulting. We have some non-sterile devices that we are working on, and we've typically cited the Code of Federal Regulations 820-130, which requires each manufacturer shall ensure that the device and shipping containers are designed and constructed to protect the device from alteration or damage during customary conditions of processing, storage, handling, and distribution. That's what we typically use for the non-sterile. I would recommend that if you have a robust packaging system and it's for a non-sterile device, you could send it through an ISTA 3 series test. If you're doing an investigative packaging system design, I would probably use ISTA 1 series, which is non-simulated distribution, but it's a more aggressive test to see what the limits are of your packaging system so that you can then make a decision whether you want to make any alterations or revisions and then put it through ISTA 3 for simulated testing qualification.
Yeah, well said,
Well,
Mike.
thank you for that additional info.
Thanks, Mike.
We have another question that relates back to dose mapping. How does dose mapping fit into the acceptance criteria? Dana? Or Jennifer, either one.
Yeah, sorry, I was trying to get off mute there. Into the acceptance criteria of the actual run itself. So one way that the sterilizer can help you, it's really about collecting data. Sometimes they put dosimeters in your product. If you have metal components in particular in your product, then they're gonna handle it a little bit more stringently. They're worried about some shielding with the radiation that's given. And really they're collecting data to make sure that every time they run it, it's consistent. And they really do that in triplicate. One way that they help customers is they do it with a continuous dose mapping. So let's say you made a product with quantity 400 and you're maxing out your over shippers in the tote at the vendor. They'll go ahead and run and collect the data on the dose mapping based on your packaging configuration, and then let you go ahead and sell that product. And then they do it 3 times. So, say 1 month you create, 400 pieces, and then 2 months later you create another 400, you'll do your second dose map, and so on and so forth. So hopefully that answers your question.
Next question is about what is your experience and success rate of implementing sterilization adoptions according to Amygar 35, and how similar does the packaging really need to be? How about you, Jennifer?
Usually, let's see, I mean, this is a little bit more outside of my normal area, because I'm just starting to learn the sterilization a little bit better. But you usually are required to do some testing just to show, like you need to have a bioburden test. Let's say you're going to do radiation sterilization and you've got a profile for one product and you bring another product in. Well, that's what they're going to be looking at is your bioburden, because that's really what determines whether or not the same level of radiation will make your product sterile. And then they'd also be looking at the density of the product, and will that density fit in with the dose map, or will putting that in with the other product cause problems? Maybe like Dana was saying with shielding, maybe you have products that have really dense areas that they don't get the same amount of penetration, and that prevents it from being really incorporated into the cycle. So you can't necessarily just say anything can be adopted. It's just like doing a family grouping, or worst case, you need to look at a lot of different aspects, think about it very thoughtfully, and write your story down and make that a very cohesive story. And it would depend, and it's not really easy to say, in this case, it would be similar enough. And there's not really a line that says, oh, up to here, I can do it. And after that, it's too different. It's something you'd have to really look at individually, and you probably would have to do some testing in order to get it in, just to make sure that it's going to fit.
Yeah, I'll add on to that, to what Jennifer was saying. So at Mainstream, we've successfully adopted in 6 products into our product family. It is based on bioburden testing as data point. That's one of them. There's quite a substantial list of things that you do have to address regarding how the product is manufactured. And then most importantly, to get into a product family, usually you have to use the same similar type of cleaning methodology and packaging, and that packaging be done similarly at the business. So for Mainstream, we offer a couple different cleaning lines, and then we do the pouching in our Class 8 clean room. And that allows a lot of, gives it a foundation for the product to potentially be part of the product family. And in addition to what Jennifer was saying about the bioburden test results. So those are the most important factors.
Thank you. We have a question about pouch sealing now. This person has had varied experience with pouch sealing machines. While they understand that the packaging design and material will impact the choice of a sealer, does the panel have a preferred vendor for sealing equipment? Anyone?
Well, I can name a few people that do have good pouch sealers. Sencorp White, and then I think they bought AccuSeal. So those brands are fairly well known. Vanderstal is another brand. I think they sell, actually the manufacturer is Fuji of the sealer itself, but those are impulse sealers. I like continuous heat sealers, I'll just say that, versus the impulse. There's different people feel differently about the two, but I've had good luck with continuous heated bars for pouch sealing. Makes it a little bit similar to tray sealing, where you've got the continuously heated platen. But there's a lot to talk about there. So maybe somebody wants to approach me after the meeting. I can talk a little bit more about it if you're interested.
Great. So there's another question in about what is your experience conducting environmental conditioning and transportation conditioning with product to be aged, or conducted this preconditioning with time equals 0?
Yeah, I can.
I guess it says,
Oh.
I'm sorry, it says age tested or only conducted at time equals 0.
Sure, I can speak a little bit to an example with that. We had a product that in previous experience got through CE Mark and was going through FDA submissions, and we had not done T equals 0 testing. We had only completed aged product testing, and the FDA said, you know, based on a worst case rationale that the longer time point was worst case. However, there was a glue joint involved in this product, and the FDA came back and said, hey, you have glue in this product. That glue is probably worst case at T equals 0 because it cures over time. And so they requested that we go back and do T equals 0 testing on all of our products. So we had to, you know, put a hold on our timeline to go redo testing on T equals 0. Fortunately, we had a lot of units already built, but that was quite expensive testing to add in at the last minute. So it's certainly something that kind of goes back to what we talked about before. If you really can justify your worst case and you do it well and it's agreed upon, then maybe you wouldn't need it, but you got to really be sure it's not always so obvious which time point is worst case. And of course, the safest path is to test both.
We always tell people to test both, just because you're more likely to pick up a packaging failure in the baseline testing, because what really damages packaging is more what's happening in the distribution test, where you're getting the interaction of the package and the product, and maybe things are rubbing on each other, they're moving around, stuff is happening. When it's just sitting on a shelf, all you have is the extra heat from aging or just time. And so that's not really where you're going to get the damage. The damage is going to happen early on. And from a time perspective, if I'm doing a validation, I don't want to wait 2 years, even if it's accelerated, to find out I have problems. I want to run my baseline set, make sure that I'm going to pass my acceptance criteria for the validation. And then if I do pass my baseline, I have pretty good confidence that I'm going to be fine with my later time points. But if I don't have that data, then I run into a situation at the end where all of a sudden I have a failure. Now I don't know, is this a failure because, you know, and then I'm stuck. And I really have to go back to the beginning anyway. So best to get your baseline data at the beginning so that you know that you can proceed. We've had lots of people who've had trouble at baseline, and they've had failures, they've had to go back and redesign. And I would sure hate, as far as timelines go, for somebody to be at the end and have to go back and start over.
And to that point, don't wait to do any testing. Don't wait to do like final validation testing or performance testing. You know, do it early. If you have a concept, or you think that something's fragile, or that it might not be, you know, a pouch might not be robust enough for it, you need to tray. Find out that early rather than, you know, build a bunch of expensive tooling and wait till the end to do, you know, 60 samples for performance testing, only to find out that it's, you know, it's not going to work. The system's not going to work. So just another tip.
Well, thank you. There's a related question here asking, do you think the FDA will have issues if additional testing is performed at real-time aging more than at time equals 0 and accelerated aging? So that's related to what you just spoke about. Have any highlights?
I think the FDA's favorite thing is when people understand what they're doing and they say they're going to do it and then they do it. And if they don't do it, that they have a good reason for it and they can tell a nice story as to why they changed course. Because things do happen. You can't anticipate when you're starting a validation that everything is going to behave as you would expect. And sometimes it would be advantageous in the middle to add in some extra testing. I think it's, if you can tell a good story and you can justify and rationalize with objective, hard fact data as to why you needed to do that additional testing, I don't think that they would kick it back.
Talking about the FDA, there's another question here about, with FDA's push to reduce reliance on ethylene oxide sterilization, what other comparable alternatives would you recommend that are as effective and worth the switch?
That's another hard one. Nobody wants to take.
You're taking the hard ones today.
Well, ethylene oxide has been around for so long, and like half the medical devices out there are sterilized with ethylene oxide. And if there was a better alternative, I'm sure some entrepreneurial guy would have come up and worked on it. There's a lot of novel sterilization technologies that are out there that are used, but they're not as scalable as EO is. So you run into a problem if you're trying to sterilize massive amounts of product that, you know, another method like, you know, the nitrous oxide one or vaporous hydrogen peroxide, they just don't scale the same way that EO will. So I think it's going to be a long time before you can transition people out of EO. I think it's going to be around for many years to come. I do think that they're working really hard on trying to reduce emissions. And a lot of that is based on EO cycles or overkill methods. They have a lot of extra built into them, and trying to slim that down more so that you're just getting what you need with a good safety factor like this instead of a huge safety factor like this. They can reduce concentrations, which reduces use, which reduces emissions. And that's probably what we'll see in the more short term. And when I say short term, I'm talking about the next couple of decades, perhaps. But there are a lot of people out there trying to come up with other sterilization methods. There isn't anyone close at this point. So it'll be a while.
Yeah, I agree with you. I think ethylene oxide is, there's nothing that comes close to having chambers large enough. And, you know, big manufacturers, they'll put pallet loads of things in a large chamber and sterilize it all at once. And you just can't do that with vapor hydrogen peroxide. The molecules don't penetrate like EO, and there isn't a good alternative right now. But like you said, people are working on ideas.
On another note, can you discuss time, cost, and device quantity requirements of both packaging and sterility validations? And can these activities be done in parallel?
That's another quite a large question.
It's another really good one, huh?
Yeah, I could speak a little bit to it. You know, just like anything else, a lot of it depends on your product, your device. There's no magic number. Certainly things can be done in parallel, yes. It's usually a trade-off between number of samples and time. So you can do things in parallel, but you might need twice as many samples because you got to build. So it depends on where you are, right? If you're a small company and building, you know, to the cost point, a lot of times the cost of the test is not really that expensive. The cost is in the number of units and building all those units and the time it takes to build those units. So often the cost is not like paying for the test itself, but in preparing the test units. So it's a trade-off and your individual circumstances will vary, right? But it's a trade-off between if you do, if you need 59 samples, it would be different than if you're doing 99, you need, 200 samples. That's going to change a lot of the calculations. So it really depends on your use case, and timing can be brought in quite significantly. Just to throw out timelines, I think you can do this kind of work in a few months if you line it all up appropriately and plan it out well. And I think, alluding back to kind of what has been said before, think it through early on. So you can probably combine and parallelize a lot of things, but think it through really well, have a well-written protocol, make sure you understand exactly what you're trying to do and what you want to do. And you document that really well so the story is clear. But also think about when you're planning a whole project like that, think about what happens if something fails. So think about, well, if I learn about a failure here, is that I've already got all these other samples on test that I've already paid for, and now they're just completely wasted because I had a failure. So kind of including all those things in the thought process about what's the most efficient for you. Maybe your samples are really cheap to make and you can do 1,000 samples, it doesn't cost you anything. Well, then, you might change your calculation. So.
And either way, you're gambling here, right? Because you have to have sterile product in order to do your packaging validation, because it needs to be done post-sterilization, because sterilization will affect your packaging. And you can't have a validated cycle until you've done your packaging validation. But if you want to be a gambling person and you're trying to save some time, you can assume your cycle will work. And a lot of times that's based on past experience. You've got a similar package that, it's not just a shot in the dark, it's a very highly educated guess. And if that cycle works out and you've sterilized your packaging validation samples to that cycle, even though it's not validated, then you would save some time. But if something happens in your sterilization validation and for some unforeseen reason, you were not able to use that cycle, now you're going to have to go back and redo your packaging validation. So again, it's a gamble, and they're not, it's hard to say cost-wise. Like for the lab, you may be looking at maybe 10 to 30,000 for a validation. But that's just money. There's still the time factor because, maybe you thought you would save a month by doing them in parallel, but because you didn't do them in parallel, now it's going to cost you 2 months to go back to the beginning.
Yeah, I agree. It's a juggling act, and it really depends on what you and your project and your company can put at the forefront. Is it, being affordable, trying to go the cheapest route? Then you probably need to apply the longest timeline so you can reuse samples on some things. And you do have to structure the entire packaging, testing, all of the things we've talked about to be able to do it in the right order if you want to try to reuse some of the samples for your testing. And then I would also add that in the discussion, we've talked about sort of worst case scenarios. Often those are the largest or the smallest and not the big sellers. And so for startup companies, it's difficult to produce all of those and then just have them sit on the shelves for a long time. So it really depends on risk, money, time, and only one of those can be the most important.
Okay, maybe Anne's microphone is not on, but this is Elisa. Can real-time aging for sterile barrier shelf stability be done with dunnage, weights in substation of product or in substitution of product? Have you seen this done for real-time aging?
Yeah, I have. It's again, it's going to be a choice based on cost and risk. You can use dunnage product. It's preferred if you don't. But again, if you have a very expensive product, you can make arguments. If you're making dunnage, you're going to have to rationalize why you're using dunnage. So give me your reasons why you can't use your real product, and make sure those are good reasons. And then tell me why your dunnage product is a representative facsimile of your real product. Is it the same? You know, maybe you use the housing and you take the electronic components out. And so then it's going to have the same interaction with your packaging, especially if your electronics weren't very heavy. Maybe you want to put something inside the housing that simulates the weight of the electronics or something. So write a good justification why you can't do it, why you're using this facsimile product. And then you may want to consider doing distribution on real product and then using facsimile product for the rest of the test. So you incorporate the real product in the part of the validation where it's the most important for the baseline samples, and then you use dunnage product. But again, it's all about telling your story. So make sure that you've got a good, clear, cohesive story about why you chose to do that and why it's appropriate.
Yeah, that's really well said. One thing to add, I think is often forgotten, is that one element of packaging often is the IFU. And so when you're thinking about, dunnage or other things, you may also want to include an IFU. It's usually cheap paper, unless you have an EIFU. But that's another example where, going a little bit above what you like, in other words, adding a little bit of a safety factor. So even if you're doing dunnage, right, you might want to make it a little bit heavier than your product in case you have a design change later where you make some change and your product gets heavier, but you could go back and say, oh, well, the dunnage we tested was heavier, or the IFU, had extra weight. And so if you update your labeling later and you add 6 pages to your IFU, you already may have some of that rationale to lean on down the road. So something to think about when you're planning things out and looking at what a worst case might be.
Excellent. There's a question about following up to ISO 11607-1 requirement that the accelerated aging has to be done within 3 months of the real time. Can accelerated aging precede real time, and can real time precede accelerated as long as the testing is within 3 months of start of the other?
Yeah, I don't think it really matters when, which comes first, just as long as they both get done and you're meeting that requirement. Again, the 3 months is essentially there because people were so excited to get their validations done that they would run out, they would do their accelerated aging, they would submit it, and then they would completely forget about the fact that they were supposed to actually do real-time testing. So it doesn't really matter if one gets started before the other. They put the 3 months in, and I would adhere to that because it's in the standard, but that's really just to make sure that the real time does happen.
Excellent. Now here's a different type of question. How much does sterilization validation cost?
So it depends on if you have to run the entire sterilization validation singularly on your entire product itself, or if you can be adopted into a product family. I would say probably ballpark figure, if you had to do everything yourself, it's probably around $10,000. That does not include dose mapping. If you are able to be adopted into a product family, then I would say it's quite a bit less than that, at least a third, just as rough numbers.
And that I think is probably for radiation sterilization, and that matches pretty much what I asked people in both of those groups what a typical validation would cost. Again, if you've got something weird going on, you could end up exceeding that, but that would be for a normal product. EO validations may be a little bit more. They were saying more like 25,000 or more, but I think overall sterilization, they're both about the same, whether you go gamma or whether you go EO. if you look at the cost to get the validation done, do the sterilization and maintain the sterilization validation over time, they usually are pretty comparable when all is said and done.
Great. At what phase of product development should packaging development be started?
Early.
At the beginning.
Yes, the earlier the better.
Yeah, don't forget about it. It'll bite you if you do.
There's actually
There's
a lot.
unequivocal.
Yeah, there's actually a lot of considerations to make at the beginning design inputs of a product that can really help with how your sterilization goes, whether you go EO or can go with gamma, including, picking out plastic components that are compatible, understanding how you may want to package it. Someone recommended looking at other suppliers, or if you have an idea of whether you want to have a tray or a pouch, depending on the weight of your product. Another example is if you want to kit 3 similar type of instruments together in one package, if you can make them a similar length, that helps a little bit in consideration. There's just a lot of inputs that either from the customer that I believe Andrew was talking about at the very beginning, or through how your product's going to be used and your product requirements. If you can include that even at the beginning, that would be the best time to do it.
And sometimes it's just really simple things. Like we were working on an instrument the other day, and I was talking with the product development guy, and we were looking at the geometry of the handle of the instrument. And there were just some really simple things that we could do there, knowing that it was going to be up against a pouch. There were some simple things, simple design changes we could make to that handle, for example, just to help us with the packaging validation. And it was easy for him to do. So just considering packaging from the start is just a good idea to keep it in mind, like what it's going to have to pass. That product's going to have to be ship tested, and we're going to use a pouch. So we better make the surfaces less abrasive if we can, or round the corners, or radius this, those types of things. It's really good to think about it early on.
Yeah, and I guess just to add the full circle of another reason why it's important to think about it early is, kind of as we've said, if you get to the end and something fails, or you make a change, as happens, or even post commercialization, you decide you want to make a design change, the turnaround time for the whole, everything that's impacted by packaging that we've talked about, some of the individual tests may be quicker or cheaper, but the scope of things that are impacted is so large and can be timely and cost expensive. So the earlier you think about it and the earlier you include requirements around packaging, hopefully the more you mitigate the risk of having huge time or cost impacts at the end or later in the project.
Thank you. Here's a question about biologic materials. Are there specific challenges in sterilizing biologic materials?
I think the answer is yes. I'm not sure, with specific biological materials, but certainly, things like E-beam or gamma can alter the certain materials, polymers or other things. They change colors or they degrade during sterilization. So I think that's also kind of true. It might affect multiple types of materials, biologic or non-biologic. So yes, there are. It just depends on what your material is. And again, going back to getting packaging requirements and including sterilization in that early on will help you to understand what your options are for sterilization, because you may actually have to change the design because there may not be a way to sterilize something if it's certain biological properties, because as we said, there's only not that many options for sterilization out there. So you got to understand if your particular material can even be sterilized as it's designed, because it might degrade in different ways.
Yeah, it's sort of the whole point of sterilization is to kill these little microbes, which are, biological creatures. And sterilization is harsh, and it's meant to disrupt the capabilities of living things, living cells, proteins, all those types of things. It makes it very hard to sterilize biologics because you can, anything that's going to kill microbes is very likely going to damage or alter biologics. So there's very limited options for that kind of sterilization. And so yes, it's hard.
Yeah, I'll just add that I guess one of the more successful methods, depending on what the biologic is aseptic filling. But you have to get the process exactly right for your material for it to work. And it does take quite a bit of development, but it is an option.
Okay, I think there's one more question posted for shipping studies. When do you do these, and how many products do you do this on? Anything regarding specific protocols?
Those can be done at any time. When we set up a distribution lab here at Nelson, it was very surprising because the market that we're in with medical device and pharma, it's such a small portion of what distribution testing is used for. And we've had a lot of problems from 21 CFR compliance standpoint, because the rest of the world doesn't care about the type of regulation that we're used to. And the people that make all this distribution equipment, they're selling the vast majority of all their equipment to people who've never even heard of regulation. So to ask them, well, is this computer system for this vibration table, is this software 21 CFR compliant? And they're like, what, what's that? It's distribution's done on any commercial product that's ever shipped anywhere, because you've got to be able to get your product to market in a good saleable condition. And it's always appropriate to do distribution testing. When we were talking a little bit earlier about pre-work, let's not wait until a validation to start seeing proof of concept that any of these things are going to work. And at any point in time, you could send a box or two for a distribution test and then send it back and open it up and evaluate it. Look inside, see if things have moved around. How does it look? And there are many different distribution cycles. When they were talking about ISTA, and you could do a 3A series or you could do a 1A series, which may be a little more stringent. And I definitely would recommend doing distribution testing early if you have any concerns at all about what your outcome may be. As far as sample size, there isn't anything set in stone. I think ISTA said something about maybe 7 boxes or something, but again, that's going to depend on what you're doing with the distribution data. If you were using it for validation, we would have you figure out how many samples you need for validation testing, however many get packed into boxes, and then make sure you have enough samples go through distribution for the end process. But if you're just doing it for R&D, maybe 1 box is sufficient. So.
Well, thank you. I don't believe there's any more questions. So I wanted to go ahead and make sure to thank all of our sponsors at TMDA, and make sure that you know that our next meeting is on April 15th. Thank you all.